Well, good afternoon, everyone. So I'm Sharon. My topic today is a review of non-typhoidal salmonella infections, and I've deliberately entitled it "The Egg, the Lizard, and Everything Else" because some people honestly do believe that you can't get salmonella if you don't eat salmon. So my presentation outline will be as follows. I'll be talking about the taxonomy, microbiology of NTS, epidemiology, pathophysiology and risk factors, clinical features, and then quick fire differences between NTS and enteric fever, laboratory diagnostics, and then management principles. So when we look at the taxonomy of Salmonella, it's actually has a very long history. It was first described from the 1800s. And it was, in fact, named after the pathologist Salmon, who was a veterinary microbiologist who was involved in the first isolation of Salmonella from porcine intestine. So that's where the name Salmonella comes from. Based on various genetic studies that have been done over the years, they've essentially come up with this very nice, concise overview of the taxonomy of Salmonella. So essentially you have Salmonella, which is the big group, and under that you have two subspecies. You've got Salmonella bongori, which is also has a subspecies under that called subspecies 5, enterica. And Salmonella bongori is rarely isolated in humans. It's named after the town of Bongor in Chad, where it was first isolated from a lizard in 1966. So it's mainly found in cold-blooded animals and the environment, and very, very few human cases have been described. The other group then is Salmonella enterica, under which you have 6 subspecies. You've got subspecies 1 enterica, subspecies 2, 3A, 3B, 4, and 6. And just to give you an idea of how big this family of organisms are, under each of these subspecies, you can have up to 2,500 serovars. We are mainly interested in subspecies 1, enterica, where again it's broken up into typhoidal salmonella, which is mainly restricted to humans, and you have Salmonella typhi, Salmonella paratyphi A, B, and C, and that's what's responsible for enteric fever. The other group is then this group called the non-typhoidal salmonella, which is not restricted to humans. You can find this in animals, you can find this in the environment, as well as human hosts. And basically they can cause a few distinct clinical entities, mainly gastroenteritis, which may be self-limiting, or bacteremia. And then you have extraintestinal manifestations, which may be bacteremia or focal infections. Of note here is that you have certain serovars that have a predilection to cause certain clinical syndromes. But obviously, as you can see, there's quite a bit of overlap between the serovars. So for example, typhimurium can cause a gastroenteritis, it can cause focal infection, as well as bacteremia. Then looking at the basic microbiology, it's a gram-negative bacillus. It measures 2 to 3 by 0.4 to 0.6 micrometers in size. They are facultative anaerobes, non-spore forming. They're motile as a result of these peritrichous flagella, and they have several virulence factors that I will speak a bit more in detail in the following slides. So its major niche is actually in the intestinal tract of humans and farm animals. So it's found in soil, in feed, in bedding, in litter, and fecal material. And so it's a source of contamination in the farms. So it's a common contaminant of vegetables, water, milk, eggs, meat products, and therefore has become a primary source of foodborne infections globally. When we look at the basic microbiology in terms of the virulence factors, you've got the virulence plasmid in the middle there, which is responsible for transference of certain virulence factors related to antimicrobial resistance. You have type 1 fimbriae, which are involved in adherence of salmonella organisms to the intestinal epithelium, and you also have production of enterotoxins, which are responsible for diarrhea. Then you've got the antigenic structures, which are quite important in terms of identification, but as well as in terms of virulence. So the first one is the O antigen, which is the somatic antigen. It's got the most antigenic variability. They're heat stable, and it's found on the lipopolysaccharide on the outer membrane of the cell wall. And you can see here it inhibits phagocyte killing. Then you have the flagella or the flagella H antigens, and this comes in two forms. These antigens are more heat labile. It comes in two forms. You've got phase 1 flagella antigens, which are phase specific, and this only occurs in a few serotypes, and so it determines the immunological identity of a particular serotype. And then you have phase 2 flagellar antigens, which are fairly non-specific, and you can find them across various strains. And then you've got the VI capsule, which is the thick capsule around the lipopolysaccharide, and that's particularly restricted to Salmonella serotype typhi, as well as Salmonella serotype choleraceus. As you can see, it prevents phagocytosis, and it inhibits complement binding. And one of the other things that it also does, and this will have relevance when we talk about serotyping, when we do diagnostics, is that because it's this thick capsule, it hides the O antigen, and so protects the O antigen from immunological detection. And one of the ways in the lab that we try and expose the O antigen is actually by heating, and that heating destroys the capsule and exposes the O antigen, allowing us to do serotyping. So it's just something to keep in mind. This slide I've just put in here just to briefly mention the type 3 secretion system within the Salmonella pathogenicity island. And really this is almost like a monster truck within the Salmonella organism that's responsible and assists with invasion, phagocytic uptake, gene expression, inhibition of apoptosis, maturation and positioning in the Salmonella containing vacuole, as well as intravacuolar survival and replication. And again, when we talk about the pathophysiology, I'll go into this a bit more briefly. So when we look at the epidemiology globally, we have a global number of cases of about 94 million cases annually, with about 155,000 deaths per year. And if you look at this map here, it shows that obviously Africa is disproportionately affected, followed by some countries in the Asian subcontinent, but really in the high income, middle income countries, you don't really see this as often. So what about the epidemiology locally? This is taken from the 2022 Germs Essay Annual Review, and this was looking at the incidence of non-typhoidal salmonella by month and year in South Africa from 2019 to 2021. And you can see that the incidence is really pretty much the same in 2019, 2021, but a little bit lower in 2020. Whether that was during the COVID pandemic and health-seeking behavior may have been contributory reasons for why we may not be seeing as high numbers during that particular period. This is looking at the number of cases of non-invasive, which is illustrated in the blue, as well as invasive non-typhoidal salmonella cases in South Africa. And what I just wanted to illustrate here is that the non-invasive cases usually have this seasonal variability. So it's higher during the earlier months, but is somewhat reduced during the winter months, and this is thought to be because of increased environmental exposure to fecally contaminated water and food during the earlier months and drier months of the year. Whereas for invasive disease, which is in the orange, you can see more or less, aside from the first 3 months here, where there were disproportionately higher number of cases in general, there's not really much of seasonal variability with that. And then looking at the serovals that are responsible, the two main ones by a long way are Salmonella enteritidis and Salmonella typhimurium murium as the main serovals that are responsible for NTS infections. And then looking at the provinces with the most cases, Gauteng is leading, followed by the Western Cape and then the Eastern Cape. And then when we look at the age group that's particularly affected, we can see that children under the age of 5 are affected, both for non-invasive as well as invasive infections. And then adults between the ages of 25 and maybe 64 are affected both for invasive and non-invasive disease. And then when we look at the primary anatomical site of isolation, really the top two are stool, which is the majority of isolates, followed by blood cultures. I wanted to put this out there because it's quite relevant when it comes to NTS, and that's really that we need to look at NTS with a One Health perspective. This was a nice paper done by Tsepo Ramatla and colleagues looking at the pooled prevalence, which was a systematic review meta-analysis of Salmonella cerevisiae in South Africa. And why I've put this here is, as I mentioned before, when we look at NTS, it really can be found anywhere. It can be found in the environment, it can be found with humans, it can be found in animals. And so we can't look at it through tunnel vision, just restricted to humans. It's just found everywhere. And so we need to be aware of that, because this has implications when we talk about IPC measures. And this is just a Venn diagram I've just put there just to show you the interaction between animals, humans, and the environment, and how they interlink with one another. Obviously, salmonella, non-typhoidal salmonella has been implicated in foodborne outbreaks, and in South Africa, the last outbreak that I could find from NTS was linked to a school nutrition program in the Western Cape, Northwest Province, where 164 children were diagnosed with salmonella after presenting with an acute gastroenteritis syndrome, secondary to processed maize meal, beans, and vegetables during their school programming. So when we look at the pathophysiology, so initially you have to have ingestion through contaminated food and water. So how much of an infectious dose do you actually need? And when you look at various textbooks and articles, the general figure is around 10 to the power 4 microorganisms that you need in order to have an infectious dose. But in susceptible hosts, between 5 and 100 microorganisms may be enough to cause disease. So once you've ingested this contaminated food or water, the first barrier to fighting off the salmonella is the gastric acid. And so the gastric acidity forms the initial barrier to prevent colonization by salmonella. But salmonella are quite tricky because they've discovered this and have now developed tolerance to moderately acidic environments, and they have what we call this acid tolerance response. So they're able to withstand the gastric acidity and then are able to subsequently pass into the intestinal lumen. So once they're at the intestinal lumen, if you look at A, they have a lot of fimbriae, if you remember from that picture that I showed you in terms of the virulence factors, that contribute to tight adherence to the intestinal epithelial cells. They invade the intestinal epithelial cells through bacterial-mediated endocytosis. That's illustrated here with the cell C. B and C are similar, except that B cells just represent M cells, which are special cells, and this is thought to be how the Salmonella typhi enters the intestinal epithelium, whereas NTS just has this generalized invasion of the enterocytes. Now, once they've invaded the intestinal epithelium through bacterial-mediated endocytosis, they then encode this type 3 secretion system within the Salmonella pathogenicity island. And basically, it's almost like this monster truck that directly injects or translocates the virulence proteins from the bacteria directly into the host cell. And what this does is it does quite a few things, and one of that is that it downregulates the host inflammatory response, but it also induces secretory response in the intestinal epithelium and initiates and recruits neutrophils into the lumen. And that also stimulates production of chloride secretion, and following that you have fluid flux into the lumen, and that may be one of the ways in which you have diarrhea from salmonella. This electron microscopy picture here is quite cool. So it's just showing as you have bacterial endocytosis, what actually happens is when the salmonella organism attaches to the intestinal epithelium, there's almost the cytoskeletal rearrangement that happens that essentially opens up the cell and allows salmonella to enter, and then once it enters, it then closes up. So I thought that was quite cool for us to see them coming through just like that. And the other, as part of the pathophysiology here, is that once you have this migration of neutrophils into the intestinal epithelium, one of the other things that Salmonella does is it actually utilizes the production of tetrathionate. So tetrathionate is, with our normal intestinal microbiome, the normal microorganisms generate toxic hydrogen sulfide gas, and they convert that to thiosulfate. In the presence of inflammation with neutrophils and reactive oxygen species, the thiosulfate is then converted to tetrathionate, which is actually used by the Salmonella for microaerophilic based respiration to generate energy. And this is one of the other ways in which Salmonella is able to effectively colonize and stay within the intestinal tract. So once you have bacterial endocytosis into the intestinal epithelium, you also have entry of the Salmonella into the macrophages in the submucosal space and into the Peyer's patches. And what happens is that you have the Salmonella containing vacuole, again with this type 3 secretion system that allows downregulation and persistence within the vacuole inside the vagocyte. And this actually serves as almost like a sanctuary site for the Salmonella to hide out, being able to evade detection by the immune system. So final clearance of the infection and immunity to Salmonella really depends on a Th1 CD4 dependent response. And you also do need production of specific antibodies by the B cells needed to control extracellular replication of the Salmonella in the blood. So this is just to highlight specifically in HIV, the problems. So the first thing is that in the setting of HIV, you have loss of interleukin-17 producing CD4 cells at the gut level. And when we have loss of CD4 mediated response, you have failure to maintain an adequate epithelial cell barrier. You have loss of antimicrobial peptides, you have loss of neutrophil recruitment, and therefore you have translocation of Salmonella. You also have cellular cytokine dysregulation and attenuation of the pro-inflammatory response during intracellular co-infection with HIV and invasive NTS. And this leads to intracellular sanctuary sites for persistence, like I had mentioned, but also it forms this site where you have a potential site for where you have reproducence of bacteremia. And this is just important to highlight because oftentimes Salmonella gastroenteritis may be very mild, maybe passing, patients may not even remember it, but even with that, you can still have Salmonella that then use the macrophage as the site of sanctuary to evade the immune system, and it can be a source of relapsing bacteremia. And this is particularly important in patients who have other immune defects, in the sense that if you have someone who has a primary bacteremia without any obvious previous gastroenteritis, you must always think of an immune defect or seeding possibly from a deep focus. And the last thing is you also have a humoral defect in HIV where you don't have effective antibody production, which is also needed to effectively eradicate NTS. So when we look at transmission and risk factors, you have foodborne factors, so live poultry or egg consumption, including intact eggshells, fresh produce, meat, fish, shellfish, nut butter, spices, flour, salt, contaminated water, unregulated herbal products, powdered infant formula, animal contacts like reptiles, amphibians, county fairs, farms, petting zoos. Person-to-person contact transmission is really uncommon as compared to enteric fever, where it is the mode of transmission. And like I said before, primary bacteremia should always raise the suspicion of an underlying immune defect or seeding from a deep focus. So really, it's not just not eating salmon. Really, everything and anything can give you non-typhoidal salmonella. And this is from Up to Date, and it's a nice table just illustrating the factors that predispose to severe salmonella infection. So if you have impaired cell-mediated immunity from any cause, HIV, corticosteroids, malignancy, if you have impaired phagocytic function, hemoglobinopathies, especially sickle cell, malaria, extremes of age, decreased gastric acidity, like I'd mentioned, gastric acid becomes your first line of defense against Salmonella, and then altered intestinal function in the setting of IBD or prior antibiotic therapy may also predispose to severe infection. So when we look at the clinical features, gastrointestinal infection is obviously, if we think about the pathophysiology, is the most obvious, has an incubation period of 8 to 72 hours following exposure. It really is clinically indistinguishable from other causes of gastroenteritis. So there's not really anything specifically in terms of their clinical presentation that can tell you, oh, this is probably non-typhoidal salmonella as opposed to the other common bacterial causes. It is usually self-limiting and resolves in 4 to 10 days. The median duration of stool shedding is 5 weeks. So that's just important to remember, because we don't often repeat the stool to check for clearance or cure. Less than 5% of patients who have gastrointestinal infections develop a bacteremia. And if the bacteremia occurs, it may lead to focal infections at other sites. And when diarrhea resolves, about 10 to 30% of adults may develop a reactive arthritis, usually of the wrists or the back of the vertebral joints of the knees. And of note, they may progress more slowly in immunocompromised hosts. So instead of presenting more acutely, immunocompromised patients may present more with a subacute chronic history of gastroenteritis. Bacteremia is the other clinical manifestations, and it may follow acute or a recent episode of diarrhea. Like I said, patients may not even recall the fact that they even had an episode of significant gastroenteritis. If it's primary, it's important, like I mentioned, to check for underlying immune defects or seeding from a deep focus. And really, the presentation here can be fairly non-specific and variable. Patients may present with fever, rigors, all the way up to septic shock. With bacteremia, you may have infection at other sites as a result. So who are the patients who are more at risk of having focal infections? It's been more commonly seen in infants less than the age of 1 and in adults more than the age of 50 if you're immunocompromised. And there is this question about if you have pre-existing vascular disease or in the presence of prosthetic material, whether you are more at risk of developing focal infections in the setting of a bacteremia. But I will delve and touch on that a bit more when we talk about endovascular infections. And like I mentioned in the beginning, you have certain serovars that have a predilection to cause bacteremia as compared to others. When we look at endovascular infections, most patients have pre-existing atherosclerotic disease. It's often the abdominal aorta, particularly the infrarenal section. And it's really based on a few case series with small numbers of patients that report that in patients who are over the age of 50 with pre-existing atherosclerotic disease, the risk of them developing endovascular infection is about 10%. So not a huge number, but definitely there are patients who do develop them. And this may also be a source of relapsing bacteremia. Often these patients present with fever, severe abdominal and back pain with or without relapsing bacteremia. And it's just important to also note that Salmonella endocarditis accounts for a very, very small percentage of bacterial causes of infected endocarditis at 0.01 to 2.9% of all cases. This is a video, I hope it plays, of a patient that I had presented, I think last year, for one of the unknown cases. And I think my marker here is showing the aorta that we're tracing down. And this was an elderly gentleman in his 70s who was a known hypertensive and diabetic, also known with atherosclerosis. And he presented with severe back pain. Cultures were initially negative, but he was actually sent to Joburg Gen because of this, a large abdominal aortic aneurysm, and subsequent cultures had come back showing that he had a non-typhoidal salmonella. So it is definitely a known complication and very scary. So how do you, figure out which patients are at risk? And really, they have come up with this non-typhoidal salmonella vascular infection score, which looks at hypertension, male gender, serogroup C1 NTS, and coronary arterial disease as risk factors. And then a subsequent study done by the same cohort was looking at the sensitivity and specificity of the NTS VI score in prediction of vascular infections. And you can see a sensitivity of about 71% of your scores less than 2 and 100% of your scores less than 1. It is also known to cause CNS infections, so usually in children and neonates, but it has been noted in adults with HIV, and this was actually in a study done in South Africa that looked at Salmonella meningitis in the South African population over a 10-year period. You can get brain abscesses, usually post-neurosurgical procedures, and also rarely secondary to a primary meningitis. You can get musculoskeletal infections, so septic arthritis, reactive arthritis, like I mentioned, and osteomyelitis, especially in the setting of hemoglobinopathies. And this is thought to be because when you have hemoglobinopathies, you may have sequestration, for example, with sickle cell, and that results in bone infarcts. And if you have a transient bacteremia, you may have seeding of that particular area, and that results in osteomyelitis. I've seen one case when I was an intern in pediatrics of a patient known with sickle cell that was then subsequently diagnosed with salmonella, non-typhoidal salmonella osteomyelitis. Other clinical features just to be aware of. So you can get pneumonia, more so in children than adults, but you can have lower respiratory tract symptoms. You can have a splenomegaly. In a fair proportion of patients with hepatomegaly, you can get a severe anemia. And I've just put this out there because, other sites are lymph nodes and urinary tract. And I've just put this out there because we've often seen patients who present to the hospital with advanced retroviral disease, not on ARVs, and they have the NTS bacteremia, and someone does an abdominal sonar as part of workup for TB. And you can see a lot of these presentations like splenomegaly, microabscesses, hepatomegaly, lymphadenopathy, anemia can all be as confounded by the NTS and may not necessarily always mean that it's TB. So it's just something to keep in mind. So this is my quickfire slide, looking at differences of NTS versus typhoid. And I've put this here because this used to confuse me a lot as a reg until I did ID and actually figured out somewhat what was going on. But really, if you look at this, I've just highlighted that in terms of NTS, like we've discussed, the host range can be very broad. Humans, animals, whereas typhoid is more restricted to humans. The reservoirs for NTS, again, can be farm animals, produce, meats, the environment, whereas for typhoid, it's mainly human-to-human transmission. The clinical manifestations of NTS is what we've alluded to in terms of gastroenteritis, bacteremia, focal infections, and then typhoid has a distinct clinical entity that occurs for about 3 to 4 weeks. They don't usually present with diarrhea because the level of inflammation at the gut mucosal level is actually quite minimal compared to NTS, and so they often don't present with diarrhea. They can have dissemination through the reticular endothelial system and actually have intestinal perforation, hemorrhage. So it's quite a distinct clinical entity compared to NTS. The disease course, like I said, is quite short for NTS, brief duration of therapy, and long-term carriage hasn't really been observed with NTS. But again, just with the asterisks that obviously in immunocompromised patients who may have symptoms that linger on to a subacute, probably chronic presentation. And then typhoid presentation, like I said, duration of symptoms up to 3 weeks, and about 5% of patients actually become long-term carriers. And then in terms of vaccination, we don't have any vaccinations for NTS, whereas for typhoid we do have the killed whole cell parenteral vaccine as well as the live attenuated oral vaccine. Yeah, so this is just a slide just to highlight the differences between NTS and typhoid because they are two completely different clinical entities. So in terms of laboratory diagnostics, you can send samples for testing in the form of blood, stool, urine, CSF, and other samples may be submitted like pus specimens, but obviously they would pick that up on routine culture. And usually it's a combination of antigen testing and biochemical reactions. So this is where we do serogrouping. So if you remember back to when I showed you the diagram of what salmonella looks like, we have these antigens, so the flagellar H antigens, the somatic O antigens, and the VI capsular antigens. And basically you have your sample here and you have antisera here, and you put all 3 of those antisera with 3 specimens that you have, and you check for the degree of agglutination. And depending on the pattern of agglutination, that will be able to tell you what serotype you are. This is the API, and this basically is another way of telling based on biochemical reactions of what type of salmonella you have. So you have all these lovely colors, and depending on what salmonella serotype you have, there is a comparison chart that you can look at, seeing if these sugars react in this particular way, etc. Then this is probably the type of salmonella that you have. Obviously, on gram stain, it is a gram-negative bacillus that's noted, and culture, it does grow under aerobic and anaerobic conditions. On selective and differential media, you have clear, colorless, non-lactose fermenting colonies, but 1% of non-typhoidal salmonella can produce lactose. And then on selective media, you can have black centers if you have indicators to pick up hydrogen sulfide, because salmonella can produce hydrogen sulfide, and that's picked up really on XLD media, agar, and they are noted to be indole negative. When it comes to management principles, I've just taken this off up to date just to give a broad idea of generally what antimicrobials we have available for non-typhoidal salmonella. It will obviously depend on your local susceptibility data, and obviously the specific susceptibilities of the specific species that you have isolated. So generally the third generation cephalosporins in the form of cephtriaxone, cefotaxime, fluoroquinolones, the ciprofloxacin, levofloxacin, and then alternative agents like trimethoprim, sulfamethoxazole, ampicillin, amoxicillin, azithromycin. But when it comes to the alternative agents, we just have to be a bit careful because of the issue of resistance. So what to do and when to do it, and this is often something that I find quite difficult to consolidate, but I've tried to make this into a flow diagram to kind of make sense of who are the patients that we need to treat and when do you actually need to treat patients. So you have non-invasive disease on the one arm, which is mainly your gastroenteritis. And if you have normal immunity, so you have patients who have mild disease and no risk factors for invasive disease, you don't need to treat them. There was a Cochrane systematic meta-analysis that was done in 2017 that actually showed there was no difference in outcome between patients who patients with mild gastroenteritis from NTS versus those who didn't get antibiotics. In fact, the group that received antibiotics had higher rates of shedding in the stool. So if you are immunocompetent with mild disease and no risk for invasive disease, no treatment is required. If you're immunocompetent, but you have severe disease, and that's really defined, again, a bit vaguely in the literature as if you have 9 to 10 loose stools a day, if you are sick enough to require hospitalization, then those patients do warrant treatment. And again, the range for treatment is 3 to 7 days. So it's quite broad. I mean, I don't really know what to make of it. Like, should you give 3 days? Can you give 5 days? Can you give 7? It's really broad in terms of the duration of treatment. And then there comes this group that if you have high risk of invasive disease, and this is patients who are at the extremes of age, and patients who may have endovascular, so risk factors for endovascular infection. So for example, if you do the non-typhoidal vascular infection score and find that they may have a high risk of developing endovascular infection, or they have prosthetic devices, then there is some debate about whether you can treat them because they are at risk of developing severe invasive disease. And again, the range here is 3 to 14 days. So it's not really clear in this particular group if you should even be treating them, but there are some papers that advocate for treatment if you are at risk of invasive disease. Then if you have lowered immunity, you treat for 14 days. And if you're HIV positive with a CD4 count less than 200, then the recommendation is to treat for 2 to 6 weeks. And again, it's such a broad range, and looking through a lot of the papers to try and figure out why is it that they've given such a big range. And what I've essentially summarized from looking at the papers is that patients who have HIV infection, their risk of salmonella bacteremia is about 20 to 100 times more than someone who doesn't have HIV. And the risk of their mortality is 7 times compared to someone who doesn't have HIV. So the recommendation is then to treat these patients, just because they are more at risk of developing invasive disease. And there's mortality associated with that. The 2-week cutoff, I think, comes from a few studies that were done in Taiwan, which had shown that if patients were treated for less than 10 days, their rate of recurrence was higher after that when they were looking at a small case series of patients. And so the recommendation was to treat for at least 2 weeks. But where the 6 weeks comes from is, again, with a small number of patients, a handful of patients, they found that the risk of relapse was about 17 to about 19% in your first month following an initial episode. So I think that's where this range of 2 to 6 weeks comes from in terms of treating patients with who are HIV positive with a low CD4 count below 200. If you have invasive disease, and that's bacteremia or focal infection, and you are immunocompetent, then you treat for 10 to 14 days. If you have a focal infection, they advocate for surgery, and then a minimum of 3 weeks of antibiotics post-operatively. If you have lowered immunity, again, treatment recommendation is for 4 to 6 weeks. And then for focal infection, again, long duration, but obviously with source control in mind. This may vary depending on where the focal infection is. So if it's osteomyelitis versus a septic arthritis versus a meningitis or a brain abscess, the duration of therapy may vary depending on where the site of focal infection is. So this is just to highlight what severe disease means. So again, 9 to 10 episodes of diarrhea, if you have high or persistent fever, if you've been sick enough to require hospitalization, and then high risk for invasive disease would be the extremes of age, patients who are HIV positive, you're immunocompromised from other reasons, and if you have endovascular disease and the presence of prosthetic devices. The role for ongoing suppressive therapy post an initial treatment regimen, for example, with patients who have vascular grafts and orthopedic prosthetic devices really needs to be individualized from case to case. So it's not clear whether all these patients actually need ongoing suppressive therapy, and we don't have a lot of data for each of the clinical syndromes. So I wanted to touch a bit on HIV and Salmonella. And obviously, this is a nice paper by Karen Keddy looking at 10-year incidence of invasive NTS in the setting of increasing ARV use. And it nicely illustrates that in patients who are accessing ARVs, your rate of invasive non-typhoidal infections reduces. But does this mean that we can potentially shorten the duration of treatment in these patients? And to answer that, this was a small study, again, of about 30 patients looking at the risk of recurrent NTS, bacteremia, and HIV positive patients with short-term secondary prophylaxis in the era of combination antiretroviral therapy. And this was looking, and basically in the summary, it was that regardless of the use of secondary prophylactic antibiotics, because some of the use of secondary antibiotics as prophylaxis was at the discretion of the treating physicians, that none of the 29 patients who are on ARVs and achieve short-term virological response developed recurrent NTS bacteremia. And I think this is something that we need to look at closely, especially because of the rise of resistant Salmonella in this day and age. So as it stands, though, the basis, so in this particular paper, patients were treated only for 2 weeks initially and then sent home on ARVs. Some were given prophylaxis, others weren't. But like I said, it didn't matter in the group that were on ARVs. None of those patients actually developed recurrent NTS bacteremia. So drug-resistant Salmonella is obviously a problem that's growing, and this is primarily from the use of antibiotics in animal husbandry, and this is where the One Health perspective is important to remember. MDR, non-typhoidal Salmonella, is defined as resistance to cotrimoxazole, ampicillin, and chloramphenicol, and XDR is where you have MDR plus resistance to quinolones as well as third-generation cephalosporins. This is the WHO pathogen priority list, and you can see NTS with fluoroquinolone resistance has made the high group. So this is something to watch out for, and it is becoming a problem. This is taken from the CDC website looking at the rate of antibiotic resistance in NTS infections in 2017, and you can see this, the profloxacin resistance is at about 10%, but also concerning this cephalosporin resistance is tracking up as well. In looking at Africa and what it looks like for us, really before 2001, we didn't have a lot of data when it came to multi-drug resistance, but really from 2001 up until now, there have been quite a lot of efforts to try and see the presence of multi-drug resistant isolates amongst NTS isolates. This is looking after 2010, and you can see that there are lots of yellows and red triangles that are popping up in Africa, and this really indicates that there's third generation cephalosporin resistance as well as fluoroquinolone non-susceptibility. In South Africa, from what I can find, I didn't really find any clear data with regards to what our resistance rates are to cephalosporins or quinolones. There have been studies done in animals looking at resistance, the NTS resistance to antimicrobials on that level, but I didn't find anything really with human cases. This is also just to bear in mind if you have patients who have obviously traveled from other parts of Africa or even the Indian subcontinent, is to bear in mind that these patients may actually have strains that have cephalosporin resistance or quinolone resistance. It's just something to always consider if the patients are not improving just because of where they have come from and the resistance patterns in other parts of the world, even though we may not have such a high degree of resistance. In terms of vaccinations, so there are a few preclinical as well as phase 1 vaccines that are in the pipeline. These are quite a few, really none have made it to phase 2 trials, but they are trying. And I think the problem here is obviously because it's across the board with humans, animals, the environment, it's really hard to find specific targets that may work across the board. But there are people who are doing intense research on this, and hopefully something will come up in the near future. And then the next question is really about chronic carriage. So it occurs in about less than 1% of patients and is particularly higher in children and those with abnormal gallbladders or biliary tracts, especially in the presence of gallstones. And this is because the gallbladder serves as, again, this little secret hideout for the salmonella to stay. They form biofilms around the gallstones, and really it protects them from the immune system as well as from the antimicrobials. It's defined similarly to how Salmonella typhi carriage is defined. So you have shedding of stool, Salmonella in stool for more than a year with positive stool cultures 1 month following resolution of the acute illness, followed by repeatedly positive cultures. Routine antibiotics are not indicated in NTS. It may be considered in select patients, and I put an asterisk there. So for example, food handlers that need to work. But like I said, use of antibiotics may actually prolong shedding in these patients and not actually help really with carriage. But if you are going to treat, the management is really then extrapolated from Salmonella typhi carriage. And then lastly, when it comes to IPC measures, obviously wash your hands properly, especially when preparing food and collecting water, when preparing infant formula, after petting animals, and cook meats and eggs well. And this is really what we can do as humans from our point of view. But remember, looking at the One Health approach, there's a lot of other things that we need to take into consideration. And that's at a farm level, at animal level, at food production and processing level, and a consumer level, which is where we come in. So really, in terms of infection prevention control for NTS, it really targets a lot more than what we can do from a personal point of view. It really does go back to what we can do for the animals and the environment, and then subsequently what we can do is really not a lot, but is going to be enough in terms of preventing invasive disease. Yeah, that's me. Thank you.
Thanks, Sharon. Fantastic. Yeah, really tricky bugs. Just going through some of the questions that have come up. So I guess in terms of the treatment algorithms that you showed, a lot of it obviously was originally based on, on gut feel, and so to speak, and more than strong evidence. Do we have? So I know you said some authorities suggested treating non-invasive disease if the risk is high or if they've got low immunity. And you gave, good plausible reasons why that, that may be important given the, the risk is really substantially higher. Do we have any evidence it helps, though? I mean, I know it sounds obvious that it would, but obviously medicine doesn't always work that way. Are there any trials to suggest benefit of treating non-invasive salmonella?
No, so in terms of non-invasive salmonella, I really looked, and it doesn't seem like in the high risk groups. Basically, to answer your question, I don't think anyone knows because we don't have a lot of trials I don't think there's a lot that has been done to look at patients who are particularly at high risk that would benefit from treatment. I think a lot of it is possible based on the pathophysiology, and it's plausible that obviously certain patients at risk may benefit, but again, I don't think there's a lot of robust evidence for that. Again, even when you read these papers looking at how long to treat these patients for, it's like 3 to 14 days. It's like a very vague duration. So I really don't, I don't think it helps. I think it makes sense if you think about it, but I don't know if I would necessarily treat someone who falls under high risk. For example, if they had severe atherosclerosis and had non-invasive disease, whether I would necessarily treat them for longer. Maybe there may be an argument, for example, in patients who are hem-onc patients on chemotherapy, transplant patients, possibly because the risk of invasive disease may be much higher in that group, that there may be benefit, but I don't think that there's actual, trial settings looking at whether it is beneficial or not. I think it's plausible, but I don't think the evidence is strong.
Okay. Oh, sorry. And then sort of related to that is this question of the duration of treatment for bacteremia. So it's really interesting. So let's say immunocompetent person, to keep it more straightforward, so not the immunocompromised. Obviously, there's been a big shift in the last 10 years away from the sort of 10 to 14-day duration and more to the 7-day duration for gram-negative bacteremias.
Yeah.
There's, several RCTs now to suggest that's fine. Those RCTs didn't have salmonella in them. One explicitly excluded them. And I do wonder about that. I mean, do you think in immunocompetent people, again, and I know we don't have the answer in terms of evidence here, but do you think that there's a good reason to keep it at 10 to 14 days rather than 7 in immunocompetent people? What do you think? I know we don't have the answer.
Yeah, I think, I'm trying to answer this in a proper way. I don't, like you said, I don't think we have an answer. I think if you look back at the pathophysiology of salmonella, even in immunocompetent patients, it does take quite a while to clear. So I think that's probably the rationale for people treating for longer, because it just takes longer to clear. And like I had mentioned, I know I mentioned this for the HIV positive, particularly the HIV positive patients, where they found that patients who were treated for less than 10 days actually had a higher risk of relapse or recurrence of NTS later on. I don't think a lot of them were HIV negative. So I think it's really based on, the retrospective data looking at more immunocompromised hosts. But I guess for immunocompetent, I'm not sure. I think, I'm not sure.
No, I mean, so I agree. So immunocompromised, definitely there's issues, especially with the cell-mediated deficiencies. I guess my gut feel with this, and it is a gut feel, and that's why I'm important to flag it, so it's not hugely evidence-based because it's been excluded, was that probably immunocompetent people 7 days would be fine. I mean, in the sense that we know this for every other gram-negative. I don't think immunocompetent people, battle to clear this particularly, but, I think it's a debatable point at least, but it hasn't been established well. It wasn't included in these RCTs, and I guess it just depends how heavily you weight it. But again, I think that's strictly the immunocompetent people. Immunocompromised, I agree. There's difficulties. I certainly wouldn't be treating someone with HIV, say, as you said, for less than 10 to 14 days. I think it's interesting and it's debatable, and I think other people on the call may feel the opposite direction. Other questions. So Kim's asking about patients who have a bacteremia from salmonella. Would you recommend that all such patients have some sort of vascular imaging or screening? Or if not, how would you triage who would get that vascular screening? Or maybe develop?
So it's quite interesting that the rate of complications where I gave that figure of 10% actually comes from, like, I think the number of patients that they had screened was about 40 or 50. So it's not a big group of patients that they're looking for. I wouldn't routinely image or screen these patients, and that's why I mentioned the NTS invasive, the non-typhoidal vascular score, which looked at your age, your gender, and if you had coronary artery disease or the presence of hypertension. And I think it's just to flag you as being high risk, and if patients then subsequently present with features suggestive of that, like if you're worried about an aortic aneurysm and the patients have severe back pain or abdominal pain, then you would image. I think it wouldn't be cost effective to kind of image or screen everybody. I think the point of that screening score is just to alert you to the possibility that this particular patient is at high risk of developing an endovascular infection, because 10% are, but there's going to be 90 or so % that don't. So I don't think it would be cost effective to like image everybody, but it's just to be aware that the possibility of a complication is there for these patients. Okay, I don't know if that makes sense. Yeah.
Yeah, no, it does. Dr. Spence is asking about whether we've seen iris with salmonella infections, and then related to that, how soon should ART be started in patients with salmonella infections?
It's a very good question, Dr. Spencer. I'm not aware of any cases of iris, but I didn't specifically look at it, so I'm not sure if you can have iris as a complication. I think in terms of starting ARVs, I would start it as soon as possible, because like I said, the sooner these patients are on ARVs, the sooner they immune reconstitute, and so their risk of the ability to clear the NTS is faster in these patients. So I would start it as soon as possible. I think after 2 weeks or within 2 weeks of them getting treatment for the NTS. I don't think there's a reason to hold the ARVs unless they've got TBM or some other reason you need to hold it. But in terms of the risk of iris as a complication, I'm not entirely sure about that.
Yeah, I mean, I agree. I don't think we've found good evidence of an iris phenomenon. I wouldn't be totally blown away if someone found a very good case series, or case report, given that they do hide in the macrophages. I guess it would be worse. But I think your key, your point is well made, that firstly, if it exists, it's very, very rare. And secondly, you really want the immune system to be reconstituting itself as soon as possible to help with the clearance, because otherwise you end up having to require this weeks and weeks and weeks of antibiotics in place. So I agree with you. I think sooner the better. I mean, so you said within 2 weeks. I mean, I'll try and get it within a couple of days if possible. And so that at the end of sort of 2 week period, you're starting to immune reconstitute. Personally, at least. Denise is asking about, within the HIV population, would you distinguish in terms of salmonella between those with a good CD4, well controlled, and those with advanced HIV disease? Would you do anything different management-wise?
I think patients who are well controlled on ARVs, you would treat similar to immunocompetent patients, as opposed to those who have advanced HIV who are not on treatment. And that's, again, we don't have a lot of data for patients who are like HIV positive with higher CD4 counts, but I think it makes sense that if you have a higher CD4 count, you are more immunocompetent. And so I think you would probably apply the duration of therapy similar to someone who doesn't have HIV but is immunocompetent, if that makes sense. Whereas those with advanced HIV, they're not on treatment. Because I think the point here is regarding the ARVs and the issue with the immune system being lowered in those patients. And that's why you need such a long duration of therapy in that particular cohort of patients. But I think it would make sense that if you are if you have immune reconstituted with a normal CD4 count, then you would revert to durations that are similar to an immunocompetent patient, because you can generate a response to that similar to an immunocompetent person.
Agreed. Yeah. And in one of your slides, you had sort of those who have non-invasive disease who to treat would be CD4 under 200.
Yeah.
you know, where you might consider doing something different, and that seems pretty sensible, I think. You know, if you're going to do anything different, that would be roughly the threshold.